Good afternoon, everyone. In today's class, we'll have a quick review of what we have learned so far from the first lesson, and then we will start learning about replication. But as always, I would like you to read something here by your textbook, and this is a nice quote. I think I agree a lot with. The greatest single achievement of nature to date was surely the invention of molecule DNA, or in general, you can make it as nucleic acids. Why is that? Because it's the only molecule with which is made up of matter, energy, and information. That information is inheritable, and that is what we will be learning. Um, how does DNA, the information that is present in DNA, how is it inherited? Quickly, um, we will review what we have learned. We learned about the chemistry of DNA, that it has nitrogenous bases, ATGC. In RNA, you would have, instead of thymine, you would have had uracil. And then deoxyribose in DNA, whereas ribose in RNA, and a phosphate group. And I hope you remember what is AMP, ADP, and ATP. It is about the number of phosphates that are present: monophosphate, diphosphate, and triphosphate. How would they be numbered? If you say this is 1, 2, 3, 4, and you have 5 here, um, so you'd have had phosphate group attached to this, which would have been P, P, and another phosphate. This would have been alpha, beta, and gamma phosphates that are attached to 5' carbon of ribose or deoxyribose. And then we also learned about Chargaff's rules, where the composition of A is always equal to T and G is always equal to that of C. Or simpler ways, the composition of purines is always equal to that of pyrimidines. Then we also learned a little bit about Rosalind Franklin's or Maurice Wilkins and Rosalind Franklin's X-ray data, that the DNA was made up is helical and it has repeating subunits. And then we have learned the structure of DNA that is solved by Watson and Crick, to have phosphates on the outside and bases inside, and that A base pairs with T using 2 hydrogen bonds, G base pairs with C using 3 hydrogen bonds. The phosphate groups are phosphate backbone is out on the outside and bases are inside. So the complementarity is one of the biggest contributions. There is one more that you need to remember is 5', 3'. Every DNA or nucleic acids has a polarity. You usually start saying 5' to 3', 5' phosphate and 3' OH. The 2 strands present you would have had in anti-parallel. Okay. So these are some of things, and we also in the last class studied a little bit about the structural dynamics of DNA, that they can go denature, meaning that they can become single-stranded DNA. If you heat it up or provide energy, then it can become single-stranded like this. And if you let it cool down, they will form double-stranded again, kind of reversible. Denaturation and renaturation. We also learned about hybridization. Molecules which have different sequences, if they base pair, then it is kind of hybridization. We also learned about the melting temperature and how GC content will influence the melting temperature. We also learned about how salt concentration in the medium or in the solution would influence the TM. Higher GC, higher melting point. Higher salt concentration, higher melting point. Some of things that we learned. So in this lesson, we will learn about the replication of DNA. I will take up Mendel's laws probably after this lesson. Let's continue with with some of the things, whatever we learned. Let's finish till at least till replication. That's a clear flow. So in this lesson, one of the important ones you should understand is about Michelson and Stahl's experiment. Then we will see about chemistry of replication, then enzymology and the mechanism of replication, and then regulation and so on. So there are multiple concepts in this particular lesson which will be so fundamental in your in the future for the future, because everything is building up, and even at a philosophical level, you are trying to see how one organism can give rise to another two organisms, something like that. So in this, you will have to, you should be able to describe the chemistry and enzymology. You should be able to explain the mechanism. And of course, there will be a few questions and like analytical thinking that you will have to do. One another important one is it's logically very stimulating when we describe something about end replication problem and how telomerases solve this end replication problem. Okay, those are some of the things that you should expect and should gain skills in. The first question is simple. What we know from Griffith's experiment or all the experiments that we have seen in the previous or in every McCarthy experiment or Hessian Chase experiments, what we know is one DNA molecule is able to give rise to several DNA molecules. How? And there is certain degree of identity. That means the sequence information that is present in the parent molecule is exactly the same in the daughter molecules. So how does this information transfer? When you don't have technical competence yet, what people do is they build models, or they draw up some model. Maybe it happens like this. Maybe it's like a hypothesis or a guess. And in that, during that time, there were a lot of people were puzzled and trying to solve it. And then there were three significant models that we should understand: is dispersive, semi-conservative, and conservative. I will take up conservative first, where there is a DNA molecule by some unknown mechanism will make a entirely new DNA molecule that is shown in blue color here. Okay. Now I'm going to describe the dispersive, where they think like DNA gets into pieces, and some parts of the pieces go into one DNA daughter DNA molecule, and some parts going into another daughter DNA molecule. That's called dispersive. What is happening to the parent molecule is the way of describing. When you say conservative, that means parent molecule is remaining as such. That is conservative here. When you say dispersive, the parent molecule is dispersing between the fragments of parent molecule are dispersing to the two new daughter molecules as shown here. And then there is semi-conservative. So what they think is one strand goes to one daughter molecule, and the second strand goes to second daughter molecule, and the new complementary strand is synthesized. At the theoretical level, anything could be possible, right? But what happens, that is how scientists work. When we don't know, we make a hypothesis, we build a model for this is how it happens. It doesn't mean we understand the whole of it or the mechanisms, but that is the starting. We will try to prove or disprove the model. If you disprove it, we will discard it and build a new model. And that is how science works. So there was fierce competition between different research groups at that time, trying to identify or find out which of these mechanisms is probably right. So there is this famous and actually an important experiment that is done by Messelson and Stahl. We'll just call it as Messelson-Stahl experiment. And they used isotopes of nitrogen. So they use two different types of isotopes of nitrogen, say N15 and N14. So one of it is certainly the 15 and 14. It's about weight, right? The number of protons and other things that it has. It is more, so it is heavy. Whereas N14, because it is one proton or electron less, proton less, it would be lighter. Okay. So they wanted to, I hope I will take a little bit of time for you to understand the mechanic, the way the experiment is conducted. So first thing, they took DNA bacterial cell, and then they grew it in medium containing N15. So N they have used is like in the Hershey Chase experiment, where they used radiolabeled phosphorus or sulfur. Here they're using nitrogen. And they have chosen N. N would be present in DNA. It would be present in RNA. It would be present in proteins as well, and many other compounds actually. Nitrogen, right? So they have taken a medium containing, say, NH4Cl, which is radioactive, N15 NH4Cl, and they have grown it in the culture. So what happens? All the molecules that are in this bacterium, they would be having radio labeled molecules. What all molecules? It could be DNA, RNA, and proteins. Okay. The whole cell, wherever nitrogen is should be utilized, is made up of N15. Okay. Now they did something different. What they did was they have taken from this culture, they have transferred it into medium containing N14. Okay, N14 medium. N14 is supposedly lighter version, right? So what did they do? Right now, this is all made up of N15. Now they put it into N14. And at that time, they knew that the bacterium divides one cell becoming two cells. I mean, one cell, it divides into two cells like this. It takes about 20 minutes, and they were using E. coli. It takes about 20 minutes for one bacterial cell to divide into two bacterial cells. So they have taken, they put it like this, at 0 minutes, they isolated the DNA from it. Okay. So although I said N15 could be included in DNA, RNA, or protein, for the analysis part, they isolated the DNA and went through it. So these molecules are not going to compromise our experiment. So they isolated DNA and they did centrifugation. The same way they have taken after 20 minutes, after 0 minutes, after 20 minutes, and after 40 minutes. I hope you understood the experimental setup. So they grew it in, they labeled, you call it labeling. They labeled the whole bacterium with N15. Then they transferred it into a different medium that is containing of N14. So from one plate, plate number 2, you can think of it like this. They have isolated DNA at 0 minutes. So they just immediately put here, and within no time, they collected the DNA. The same way, they have put in one of the mediums, and in about 20 minutes, they collected the DNA. In the third plate with N14, they collected the sample in 40 minutes. Now you have 3 different DNA samples, right? And they used analytical centrifugation for it. So I hope you remember from analytical techniques, you use centrifugation for two purposes, mostly for preparative purpose, but you can also use it for analytical purpose. They took a tube and they created cesium chloride density gradient. Okay, that means you will have high density, uh, and as it is growing, you will have less density. The N15, whichever is very heavy, will settle faster, and because it will also be dense, it will settle fast. Whichever is lighter will travel slower. Okay, that is how you would have the DNA to do. So if you take, let me check here. I don't know if it is continuation or not. Okay, if this is labeled parent DNA, that means which is containing N15. Okay, both the strands are N15. It would be very heavy, so it will travel. It will settle, or it will settle much lower. Whereas if it is unlabeled DNA, then it is usually unlabeled one is the N14. So you have just taken DNA from a normal growing bacterial cell and you have put it in, you centrifuged. It will sediment to at this stage, at this place. Assume that. But if you have labeled with N15, which because of which it is much heavier, a little heavier, it will settle here. Okay, there is a difference in the place of how they are sedimenting. Okay, so why should it? There is a centrifugal force applied, and because of it, the molecules, if they are very heavy, they will tend to move down faster. If they are light, they will be somewhere here, something like that. So when you say F1 generation, this sample is after 20 minutes. This is after, you can count it as 0 minutes, okay? And this you can take it as 40 minutes. What is happening? If it is at 0 minutes, both the strands are N15, made up of, or labeled with N15. After 20 minutes, if I go back, you notice that after 20 minutes, what we are doing from N15 medium, we are putting in N14. So any bacterium that is growing would have used the nitrogen that is present in the medium. And that medium, that nitrogen present there is N14, lighter version. So the DNA showed a band in between that of, if you see, unlabeled one is here, labeled one is here, right? I will erase it and then I'll get back. So I can draw a line like this. You can put it this way. That is unlabeled. This is where is labeled. And this band was formed in between like this. What does that say? It must be a mixture of parent DNA that is containing of N15. It is between the bands of, this is N15 band and this would have been the N14. Somehow its weight is between that of N14 and N15. So people thought, which of the models can you eliminate? We had three different models, right? One is dispersive. The other one is semi-conservative. The other one is conservative. If it were conservative, what would you expect in this stage? You would have had one N15 band and one N14 band like this. If it were dispersive, that means you might have a band like this in between. It would have, all the daughter DNA would have been a mixture of parent DNA as well as newly synthesized DNA. If it were semi-conservative, you would have a band like this. So at least we could have removed away conservative model, because these two bands are not formed like in here. In this case, you're forming one band and it is between the molecular weights of N15 and that of N14. So you can eliminate conservative model. Now, At 40 minutes, what is happening? This is at 20 minutes. At 40 minutes, if you have, what should happen is the question. If it were dispersive, what would you expect? You would still find only the middle one, right? Little shifting towards this one. So something like you would not find, okay, what is found is what we will see. They found one band showing that of, which is a hybrid between N14 and N15 type molecule, and the other one is completely N14. Why is that happening? We will proceed as we go ahead. We will see some more examples where your understanding would be better. But I want you to think of it in analytical ways, not just mug it up. Okay, it's easy to mug it up, but it's not good for scientific training or thinking. You should try to understand, be in the shoes of the scientists and see what could have been there instead of directly getting to what is the point of it. So this is how their results would have actually would have. These are their results actually presented. This would have been the molecule that is made up of N15 only. This is between. This is the one a hybrid that's made up of N15 and N14. And in round 2, you're finding a little bit of this, but a new band that is completely made up of N14 was formed. If it were dispersive, you would not see two clear bands like this, isn't it? Say, what is happening in dispersive? You will have these parts cut into pieces, and I'll just make up some. I hope. Just give me a minute. Okay, so if a DNA molecule is made like this in the first round, that is where something like dispersive, okay? Just think of it. And this would have formed because each of the DNA molecule is made up of hybrid N14 and N15, so it would give a brand like this. What would happen in the next round? In the next round, again, you will have a few of them. I'll draw 4 molecules, okay? Each for something like this. I should not draw more of N15. I should keep that in mind. How many are there? 1, 2, and something like that. Okay, the other one could be this. So basically, what you're seeing is the chances of forming a two distinct bands like this would not have been so possible. Here I have drawn only two fragments. DNA is very long, and if they were, each of the molecules would have gotten a little bit of N15 wherever it is there. So they will all form one diffused band, something like you will have a diffused band like this, not the clear bands as shown here. Okay, from that interpretation, what is possible is that it is semi-conservative replication. You read upon it. We will again, when we will review this, and hopefully you'll find it logically put. So this is the same thing where we are now seeing it as an example of how things happen. N15 bacterium is the DNA is labeled, and when you transfer it to N14 medium, you have semi-conservative, one strand made from N14, one strand made from 115. And the other one, if you continue to grow, say something like 40 minutes, this is at say 20 minutes, and this one is at 0 minutes. And all the new ones that are being made will only have N14, right? So DNA is isolated and allowed to with cesium chloride solution and allowed to centrifuge, ultracentrifugation. Okay, it's not density gradient, or it is, yeah, it should be gradient. Anyway, so when you centrifuge, the heaviest ones will settle farther, from the center of rotation, and the medium ones will be somewhere here, and the lighter ones will be much more closer to the axis of rotation. So you should understand also the why is it called ultracentrifugation, and for how long did they do it. The difference molecular weight between them is not too different, too distant. That's why they have to use cesium chloride. They have to use very high centrifugal force and for a long duration to get something, a result that was shown in the previous slide. Okay. So I hope you understood this thing, right? Heavier one, that is N4, N15, N15. This is semi-conservative replication, which is a hybrid between N14 and N15. And in the next round of generation, you would find formation of N14 and N14, while a little bit will be the same. I might ask a question like this, that what would have happened if dispersive model is correct? What would you expect in after one generation and two generation? This is for scientific thinking. What we know now is that semi-conservative replication is the correct one. But the questions when we ask, it could be like, can you draw a representative diagram of the results that would have obtained if dispersive model is correct? Or a question like this would be, what would have happened if they went for further generations? This is one generation, two generation. If they went for the third generation, what would you expect the bands to be? How would you think they would look like? Okay, that is involving scientific thinking and critical thinking. Okay, what we know now, we will get into the chemistry of it. There are two substrates for the DNA synthesis. That is what we are trying to understand, the semi-conservative replication, which is the actual way of how DNA replicates. And I hope you remember the bases, the deoxyribose, 2' H, and then alpha, beta, and gamma. So DNTPs, that is deoxynucleoside triphosphates, they are the building subunits, okay, substrates. There is also another substrate, you call it as primer template junction. What is a template and what is a primer? Template is something using which a new something else is made, right? You know about a mold. If you take a mold like this and you put in, you must have played with the clay. You put in the clay, you will generate something like this, right? So this mold, this is the template, and this is the new thing that you made out of it. Okay, same way you need a template. And what is the template used for? The bases that are there through which hydrogen bonding can be made is what is used for. So the incoming, if you have A here, you know what base should come in there. That would be T. If you have G here, it is C should come in there. So which base should be added here is determined by this template, by this strand, and that's why we call it as template strand, at least in this picture. There is one more problem, is all of DNA synthesis is in biological system is determined or is catalyzed by an enzyme, and that enzyme is called as DNA polymerase. In short, you write DNA pol. So the DNA polymerase is an enzyme that adds bases according to using the template. Okay, if it finds A here, it will add a thymine here. If it has T here, it will add a adenine here, something like that. It reads the template and it polymerizes the bases sequentially, one base at a time. The one problem that DNA polymerase has is if there is a DNA molecule like this, It cannot come and start, um, say if there is a DNA molecule like this, it cannot just come and add here. It needs a substrate, that is, it needs a OH group. And only to the OH group, again, I have to answer. Here you see this 3' OH. DNA polymerase can only add more bases to this 3' OH. And a new one will have there will be a phosphodiester bond formation, and you have this, and there is a new 3' OH. So DNA polymerase can only add molecules to this, add bases to 3' OH. It cannot it cannot initiate replication by itself. So there should always be a small length of DNA like this, which is because there is base pairing, you have double-stranded DNA, and the rest of it is single-stranded. And this stretch of nucleic acids, which supplies or has the 3' OH, is referred to as the primer. As a part of practicals, we will also do PCR, polymerase chain reaction. So I hope you are understanding things properly. Please read the textbook, watch the videos on YouTube. Also, there are a lot of videos. I would want you to be enthusiastic about it. And there is a fantastic video. I must post it soon. I will do that to better your understanding. Okay. So there are two substrates. It is DNTPs, and the other one is primer template junction. Template is the one which is whose base sequence is red. Primer is the one that supplies the 3' OH, onto which DNA polymerase adds more bases, one base at a time, using the complementarity of the template. So mechanism, or simply what happens is the base pair T is here, so it can base pair with A. It cannot base pair with C, G, or another T. It can only base pair properly with A. And then this 3' OH that is present will make a nucleophilic attack onto the alpha phosphate, and then these two phosphates, that is beta and gamma, are released out, and this pyrophosphate is further hydrolyzed to inorganic phosphates by an enzyme called as pyrophosphatase, and this phosphodiester bond is made here. All the energy of breaking the phosphate bonds is utilized in making the in polymerizing the DNA here by DNA polymerase. Okay. So this is the role of 3' OH. Is the DNA polymerase will activate this 3' OH, or it will actually deprotonate it. So this is gone. You'll have negative O, which will then make a nucleophilic attack on the gamma phosphate, and then add this base here. So for mechanism, if we have to really understand, we have to get into the enzymology and how DNA polymerase does all these things. Say, for example, how does it figure out what base is here? That's the first thing. Second thing, how does it figure out that the base pairing between the incoming nucleotide and the base on the template is accurate? Okay, so DNA polymerase, like many other enzymes that you will read here, is very accurate, and it's a fantastic one because it has the capacity to even correct if there is anything, if it made a mistake. It is a self-correcting. Okay, how many of us have this property? We tend not to look into our own mistakes often, but DNA polymerase does have capacity to find most of the mistakes, if not all, or it has proofreading mechanisms. The word that we use is proofreading. Okay, that allows DNA polymerase to make any corrections if it had made any mistakes. So we will look into some of the things today, and then I will continue. I will review and continue in the next class. The first thing is I wanted you to have this. Okay. What it does is this is the template you are finding. This is the template is straight put like this. Okay. This is the base. First question we are asking, the point that this slide is actually showing is you have D and TPs in the cell. There are also NTPs. How does DNA figure out which one to use? The difference between these two is just one oxygen molecule, right? Say if you took ATP and normal ATP, the difference is just in the whether it is oxyribose or deoxyribose, just one oxygen atom, and then DNA polymerase should be able to discriminate. The problem is also that the, the concentration of DNTPs is very high compared to that of DNTPs. In that situation, how does DNA polymerase recognize? I mean, it should not take up ATPs and should only bring in DNTPs to be polymerized sequentially like this. So for that, what you're seeing in this image is this is the template, and here is the primer. Okay, so I'm showing 3' OH. So this one should have been the template should be 5' to 3', and this one is in this direction, 5' to 3', and this is a new base that is coming, new nucleotide or DNTP. Okay, the difference between figure A and figure B is just that OH here. If there is a region within DNA polymerase that is referred to as that has discriminator amino acids, if there is a OH, they cannot make an proper interaction. This is RNTP, ribonucleotide nucleoside triphosphate. If there is OH, like in that would have been present in the 2' ribose, these discriminator amino acids will not allow the interaction to happen. That is nucleophilic attack, activation of 3' OH and making an attack here. That is not allowed. Okay, so the RNTP would have been eliminated from the active site. If it were just DNTP, which doesn't have this OH here, then there is no hindrance by the discriminator amino acids. Therefore, the 3' OH would be activated and to be made um, to make a nucleophilic attack at the alpha phosphate, and then this beta and gamma phosphates are released out. I hope you understood. Even though the concentration of DNTPs is very less compared to that of NTPs, even though the chemical difference between NTPs and DNTPs is just one oxygen atom, still DNA polymerase can recognize or differentiate between what is RNTP and what is DNTP. Thereby, it will only allow the entry or make incorporation of DNTPs into DNA and eliminate any RNTPs that may have entered into the active site. Okay. So there is this is typically how DNA polymerase is seen or represented for the sake of understanding. You can take your right arm and close it like this, and on one side you would have thumb, and the other 4 fingers would have been on the other side. The palm region that you have here, that is where the DNA is. So there is also one more question, right? If you have a primer and template, and here you have one base, second, third, and so on. How does it know that this is where, opposite to this, we should have added a base? Why not it add a base here or here? That is how that base is brought into the active site is the point. So if you see in this picture, you see a double-stranded DNA. The green one is the growing chain, or that is being polymerized, polymerizing strand, and the gray one is the template. Okay, so if you follow the template, the gray one here, the only one base is exposed here. The rest of the template is outside the active site of DNA polymerase in the palm region. That means that is how DNA will figure out that a nucleotide DNTP would be taken here and added to that particular base and not to any other base in the DNA, in the template region. Okay. Um, yes, I'll take this and I will stop with that so that I can go for review. There are some concepts that we have learned which might be critical. So how does it activate the 3' OH? That's the question here. There are two metal ions, Mg plus. One is referred to as metal ion A, and the other one is Mg2 plus, and the other one is referred to as metal ion B. In the active site, there are these these metal ions that are present. This metal ion is used or activated to deprotonate. Okay, this hydrogen is removed, because of which now it forms an oxyanion, and that oxyanion will make a nucleophilic attack on the alpha phosphate. Okay, so what you should remember is these two metal ion A and metal ion B are important. What is the function of metal ion A? To deprotonate 3' OH so that it forms an oxyanion. And the second one, metal ion B, it actually keeps the alpha, beta, and gamma positioned appropriately. If there are negative charges that are kind of mitigated by the metal ion B. Okay, all this would have happened if the discriminator region is allowing the. There are two things here. One is that the hydrogen base pairing should be correct for AT or GC interaction. First thing is hydrogen bonding should be good. Second thing is that it should not be a ribose. It should be a deoxyribose. When these two are fine, then everything is structurally in a position where deprotonation of 3' OH can happen here, formation of oxyanion, and then metal ion B will keep this alpha, beta, gamma in this kind of arrangement. So the oxyanion will make a phosphorester bond here. Okay, there will be more things that we will discuss and I will review. And if you have any questions, you can quickly ask.